is Steve Prager. I am the director of the Appleton-Whittell Research Ranch of the National Audubon Society. We are a roughly 8,000 acre field station Audubon Center and wildlife sanctuary here in southeastern Arizona. And here we put these grasslands to work, answering the questions that are most critical to us achieving our mission of protecting birds and the places they live and all of the other critters that share those spaces with them, including ourselves. So before I pass it off to our speaker today, I do want to talk to y'all a little bit about our Appleton-Whittell Research Ranch fellowship program. This program was launched in 2010, and it supports up to 3 research fellows like Marcus every year. Our research fellows are young scientists between the ages of 18 and 30, and they are focused on conservation-related research, questions that can be answered here at the research ranch, and that show a strong conservation ethic and a commitment to sound science. Each fellow receives a $1,000 cash stipend to support their research needs, a $500 Appleton-Whittell Research Ranch housing voucher, which supports about 2 weeks worth of overnight stays here on the ranch, and communication support from Audubon Southwest to help these students, these researchers, get their stories and their research out to a broader audience, which of course is exactly what we're doing here today. The application window typically opens in early February and closes in late March. So if you happen to know a young scientist that might like to do some work on the research ranch next year, keep an eye out early in the year for that announcement, and we would love to hear from them. So what is keeping our research fellows busy this year? That is a question I am going to pass off to Marcus. He is one of our fellows this year. Is an undergraduate from Christopher Newport University out in Virginia, focused on studying ornate tree lizards. With that, Marcus, I think I will stop sharing and I will pass it over to you.
Terrific. Okay. So, hello. My name is Marcus. I am an undergrad student at CNU, like Stephen said. And the question that we're kind of investigating today is the eyes of 3 to regulate UV question, whether the parietal eye is related to UV regulation or some other role in the ornate tree lizard or Urosaurus ornatus. So Stephen gave a wonderful introduction, but just a little more about me to kind of set up how I approach this topic is I am from Northern Virginia, but I do attend Christopher Newport kind of in the southeastern part of the state. And there I've looked at a lot of amphibians and reptiles and dabbled in some estuarine fish. And those serve as really good organisms to look at all kinds of traits like in their ecology, life history, and evolution in ways that can be applied to some broader conservation topics. So even though these organisms might seem like a random kind of grab bag of thing that don't make a lot of sense to the uninitiated, there's a lot of ground that you can make up with applying these types of questions in that type of way, which is similar to this kind of random sample of objects here. So we have rubber cement, we have tape, there's aluminum, small scale surgery, or at least my poor clip art representation of it. And just keep in mind, I'm hoping you'll take a few seconds to kind of ponder what these items could possibly have in common with each other. If you want, if you're really dedicated, you could type it into the chat. If you want others to know how your line of thinking is, because this is truly a very random seeming mixture of objects. So a few more seconds, and then I'll give the answer in 3, 2, 1. So all of these were applied to lizards' heads, not all at once, in some different combinations. But each of these items has made its way onto the center of the head of a lizard. And you're probably wondering why, because this is an odd thing to do for sure. Some of you might have remembered back from the title that I mentioned a structure called the parietal eye, which is in the center of lizards' heads. And these structures were essentially used to help scientists research that structure and what it does to better understand these lizards. But what is the parietal eye? So essentially, it is a photosensitive structure that is capable of detecting light, as that's what photosensitive means. Um, and it determines the intensity and direction of light. So if you look at the diagram on the right, it's very similar in structure to the lateral eyes, or the eyes you're probably more familiar with. But it is not near as complex. It has just the same basic format of photoreceptors on the bottom, a cup shape, which helps it determine direction, and a lens on top to focus the light. Um, and so it does not form as complex pictures as your lateral eyes would, because it doesn't attach to the brain via the optic nerve or the kind of visual processing sensor. It attaches to the brain through the regions of the brain associated with thermoregulation and circadian cycles through the production of melatonin. So these are kind of the processes that we look for that it may influence within lizards. And I would just like to clarify that it does exist in other vertebrates as well. So they have been observed in some fish, like the paddlefish. They have also been observed in amphibians, like salamanders and frogs. Um, and it was likely present in some early vertebrates as well, though there's no fossil evidence of it, and it's just largely theorized. And the vertebrates that don't have it would include turtles, birds, crocodilians, mammals. Essentially, those groups have the parietal eye kind of absorbed into the pineal gland, that region of the brain I was mentioning that kind of controls those functions. So there's no outward extension of it into the environment. But for the sake of this presentation, we're only going to be worried about lizards. So to provide a kind of brief history of the research of the parietal eye, in general, these previous studies have focused on relatively large species of lizards. So the collared lizard is the smallest one here, but they can approach 10 inches to a foot long. Bearded dragons are a little over that, and monitor lizards are well over that. And these studies have opted to test the function of the parietal eye by either surgically removing it or blocking it with a aluminum tablet and an adhesive. So the adhesive was either tape or, in one case, rubber cement. Um, and these trials in these experiments took place over relatively long periods of time. I say relatively because, as you'll see later, the trials we did for our experiment were only an hour, whereas these studies wanted to see how the parietal eye impacted behavior for usually an entire day. And they did that because they were interested in the circadian cycles and full day kind of thermoregulation process rather than just temperature preferences or UV preferences. And so to kind of break down these studies a little further, one study that focused on collared lizards had removed the parietal eye and found a insignificant trend that resulted in lizards choosing higher body temperatures. And when you remove both the parietal eye and the pineal gland within the brain, lizards did significantly select lower temperatures. So this is just kind of a further demonstration that there is some interaction between the parietal eye and those regions in the brain, and that it does influence their kind of regulatory homeostatic behavior. A further study in collared lizards had demonstrated that the average temperature that they selected over a period of 24 hours was not significantly different when the parietal eye was covered or left exposed. But during the scotophase, or their the like nighttime period, essentially the period of dark hour when the lights were off, it was simulated. They selected higher temperatures at that point. So not enough to come to any kind of conclusion, but the parietal eye is likely doing something in regards to their perception of their habitats. The research on monitor lizards focused on two species, the Asian water monitor and the clouded monitor. And both species had lower locomotion periods when the parietal eye was covered. But only one of them had ended up changing its selected temperature. So the water monitor selected lower body temperatures when their parietal eye was covered, whereas the clouded monitor had no difference in the selected temperature. And then more recently, a study on bearded dragons essentially covered the parietal eye with aluminum and wanted to investigate the impact the parietal eye may have had on weak electromagnetic frequency fields. And essentially, they tracked how often they did a tail lift response, which you can partially see in that top image. And essentially, when lizards were exposed to the electromagnetic field, they would lift their tail more frequently than if they weren't, unless their parietal eye was covered. So the kind of what those results would indicate is once the parietal eye is covered, they're unable to detect that field as strongly. And so in conclusions from these literature, we can see it likely influences some aspect of their diel cycles or their daily cycles, their thermoregulation, and detecting the electromagnetic fields and spectra. And it operates with the integrated brain regions that it's most closely associated with. But at a broader scale, the function of the parietal eye is still relatively mysterious and not very clearly known. But thermoregulation has remained one of the major hypotheses for what it has to do with. And that's not for bad reason or anything. It's a good theory to follow because lizards are ectothermic, meaning their body temperature matches that of their environment. And so detecting metrics of how the temperature would vary within the environment is an important thing to do for them to be able to maintain their kind of internal environments since they can't control it with metabolic processes the same way mammals and birds and other endothermic animals can. And so essentially temperature is important because it determines their digestion and metabolic rates. The enzymes that break down their food have an optimum temperature they function at. That in turn determines how much nutrients they can acquire, which determines how fast they can grow and determines how much energy they can have, which can determine their activity levels, which would be their ability to forage for more food or their ability to escape from predators so they don't become food. And all of these factors contribute to the overall survival and fitness of the lizards. And so that is largely why temperature and thermoregulation has been a focus of lizards for so long. And this behavioral thermoregulation has been kind of contributed into two categories, namely basking, which is where lizards will sit out exposed to sunlight in an attempt to gain more heat from the sun and warm up their bodies, versus shuttling, which is where they'll move between shaded and lit areas where they might be foraging, they might be hiding, they might just be moving from a more hot spot to a less hot spot throughout the day. However, as we had shown earlier, the research hasn't provided a strong consensus on how the parietal eye affects thermoregulation. And that's largely just because of variance in the time periods those studies were following, variance in the species those studies were following. They're not very closely related, even though they're all lizards. And in general, just the different methods of inhibiting the parietal eye, whether it was surgery or an adhesive or what type of adhesive, those are all things that can be taken into account that kind of throw off the integration ability of the research. And so in more recent research, it's become a bigger focus on how heat and temperature is not the only determining factor for reptile behavior. They don't just need to regulate their body temperature to maintain homeostasis. They also have to regulate their exposure to ultraviolet light or UV. And the importance of UV cannot be understated. So when we're talking about UV, essentially on the electromagnetic spectrum here, a little shorter frequency than the visible light spectrum. We can't see it, but some animals can. We'll get more into that later. Namely, the UVA and UVB spectra are going to be important for reptiles as they get through the atmosphere in the greater quantities. And so UV is incredibly important for them to receive because it stimulates their production of vitamin D3. So they have a cholesterol in their skin that, when exposed to UV light, cascades through a reaction to where it becomes pre-vitamin D3 and then will become vitamin D3, which is a very important vitamin because it is required for calcium absorption. So if you have pet reptiles, or you're familiar at least with the kind of reptile trade, you've probably heard of metabolic bone disease and the importance of providing UV light and calcium and vitamin D3 to reptiles. So you're probably familiar that if the UV light is too low and they can't get vitamin D3, then they can get metabolic bone disease. But in an ecological context as well, they can have a lower reproductive output. And that's another kind of cost that would affect their fitness and overall success in the environment. Conversely, if the UV is too high, they can get skin and eye damage, usually in the forms of burns or blindness. And they can also get hypervitaminosis D, which is essentially just too much of that vitamin, which can throw off their homeostasis just as much. And each species will have an ideal range where they can maintain their homeostasis of UV, where they can have healthy organ function, healthy bone development, and healthy development of embryos during their reproductive season. And so this kind of range is what every lizard is trying to achieve in regards to their UV exposure. And so a lot of the work regarding what ranges different species select has been done by a scientist, Gary Ferguson, who had kind of divided the UV index into 4 different zones named after him, Ferguson zones, and different lizards, respective to their different habitat use, will occupy or prefer different zones, different Ferguson zones within the UV spectra. So a crepuscular lizard like the leopard gecko that doesn't experience much UV at the time when it's active will logically and empirically through research select a lower UV value for its preferred range, whereas the horned lizard that lives in the more open, sunny, arid deserts during the day that would experience greater UV has a higher UV preference. And this is essentially the context in which the Latanzio lab, which I'm a part of at CNU, attempts to study UV in this kind of ecological context of what UV levels do they prefer and how do those trade-offs influence their kind of ecological behavior. So essentially, with regards to kind of challenging that history of assuming lizards do everything because of temperature, if you gave lizards their preferred UV but at higher temperature, how far are they willing to risk going to their maximum temperature in order to get their preferred UV? Or if you have a species that occurs at high elevations where UV is high but temperature is low, and also at low elevations where temperature is high and UV is low, then how does that influence the thermoregulation and UV regulation of that species, depending on where it is in that habitat mosaic? And so in general, while we're collecting data of where they are temperature and UV wise in the field and collecting just general habitat data and where certain trees and microhabitats get certain temperatures and UV in the field, a big component of this also involves bringing lizards into the lab, into controlled arenas, and seeing what temperatures and UV they prefer when all other costs of predation or the need to find food are removed. So these arenas are essentially used to provide a gradient from low temperature to high temperature or low UV to high UV and provide that kind of choice or options for the lizards to select and show us what they like so we can measure how well they're getting that in different habitats and under different pressures. And so those kind of trials are the things that I've generally helped out with the most within this lab. And one of the most important parts of running those trials is this structure, this thing on the lizard's back right there. So that's the sensor that we attach to the lizards to actually detect the UV. The UV sensor is that square with the shiny circle in the center of it. And the important part of that device is it has to be on the lizard's back pointing directly vertical in order to actually detect the UV level. If it's tilted off to the side, it doesn't read the UV coming from the light in the arena. And the reason this kind of caught my attention is that's very similar to the position that the parietal eye is in on the lizard. Of course, one's on the back and one's a little further up on the head, but they're both kind of dorsally situated in this way. So the parietal eye might just be very similar to this technology that we're sticking on them to sense UV. So we have a photosensitive structure that's cup-shaped, meaning it can determine the direction of light and therefore which side would have higher UV or lower UV, depending on how the rays are coming in. and it's dorsally situated, which is often where lizards will have their back to when basking. They'll have their back to the light source providing it. And we know it has responses to weak electromagnetic spectra and homeostatic regulation behavior. And so essentially, this led us to the question of, is the parietal eye used for UV regulation in lizards? And this is a bit of a tricky question to answer just kind of in a vacuum like this. So overall, we kind of can break it down into, if you cover the parietal eye, does that impact lizards' ability to regulate UV? And so going into this kind of procedure, we would have expected either if you cover the parietal eye, then the lizards will have a greater difficulty reaching their preferred level of UV. So that could come in the form of either when it's covered, they on average select a different value than their preferred UV. Or maybe they arrive at the same value, but there's a lot more variance in that average. They're not as precise with their selection, not of the UV value that they prefer. Alternatively, it is possible that there is no effect on their UV regulation if you cover the parietal eye. And that could mean, and that's not as simple as it sounds at first either. So it could mean the parietal eye doesn't detect UV at all, or it could simply mean that the parietal eye is not the only thing that detects UV. And that if you cover the parietal eye, there are still other UV sensitive sensory structures on the lizard that can pick up the slack when one is lost. So their lateral eyes can also detect UV. The cholesterol that gets converted to vitamin D3 is in their skin. So it is plausible that potentially there's just some kind of reaction they can feel within their skin to sense where UV is. But these are kind of the main two hypotheses we had going forward with the experiment. And so essentially, our study species for this project was the ornate tree lizard, Urosaurus ornatus. So this species is much smaller than the previous species used in parietal eye research, which it's not particularly closely related to either. It does share part of its range with the collared lizard, but overall, it's quite different from the previously studied animals. So there was some different thought we put into the selection process for this species. And the main reason it was our top candidate was because they are abundant and they are widespread kind of habitat generalists. And that's important for us when considering gaining a large sample size. So the previous research that had larger lizards also tended to have smaller sample sizes just because it's harder to keep more lizards in a lab-controlled environment when they're bigger and you need to give them more space to watch how they react to the different environments you present them. And we also have studied these lizards in the Latanzio lab previously, and so we have demonstrated through those kind of studies that they are behavioral regulators of both temperature and UV. And so if they are under pressure to select, to sense these environmental factors and respond to them accordingly in that kind of fashion. And as a kind of a side note, just a kind of fun fact about them, they do also have polymorphic coloration. So the males are depicted here. They can have throat colors ranging from blue to yellow to orange, or some combinations of two colors. The females are generally just strict yellow or strict orange or white under the throat. But these are correspondent to their reproductive strategies. So the males will kind of stilt up their bodies and do push-ups while displaying out that throat pouch to kind of attract females. And that's a kind of a bit of an aside for now, but it will come back later. We'll get there. And so essentially, with this in mind, we have this abundant lizard with a wide distribution. And so the next step, once we have that species selected, is the field work needed to collect that species. So this is where our partnership with the Appleton-Whittell Ranch came into play. So the My professor, Dr. Matt Latanzio, has studied the ornate tree lizards on this site for quite some time and has some sites picked out for where they are kind of most abundant. And the ranch has a good bit of different habitats that they are known to use. So these live trees, like you see on the left, or more of the snags and dead trees that you see on the right, are all habitats they'll utilize, and that is in no shortage on the ranch property. So that's where we have had success collecting them. And to collect them, we use the kind of traditional method of the catch pole with a loop on the end, just because it is the most safe way of catching them without risk of injury to their limbs or causing them to drop their tails in any way. And so in order to do this as well, you also do have to be able to find the lizards, which is arguably the most challenging part. Since they do have such cryptic coloration, they blend in really well with that bark on the trees, whether it's dead or alive. So these ones here with the white dot on their tail, that is a spot of whiteout we give them upon release so that we don't catch the same lizard twice. I used them for these photos so that I could promise myself I would find the lizard again to show it on the slide. Um, but generally, you'll see them move. You might catch them displaying, you might catch them foraging, or if you just notice a weird smudge on the bark that kind of doesn't follow the same path as the rest of the bark, that's usually the typical ways you'll find the lizards in their habitat. And so once we kind of have them collected, we'll bring them into lab and we'll house them in these plastic critter keepers or totes with a hide and their full spectrum lighting provided. So they can have their heat and have a refuge to get away from that heat to thermoregulate properly for their benefit of their health. When they're not in the trials we kind of situate them through. And so they were also provided food for the days that they were not, for the days that did not conflict with when they would go into a trial. So food is a confounding variable for temperature and UV regulation studies, since those kind of environmental factors are important for how they'll process their food. And so we would only provide food on days where it wouldn't proceed a trial. And once we had them in lab, we would also collect some information just on their general morphology. So we would determine the sex of the lizards. The males will have these long blue bars on their ventral side on their stomach, and the females will generally be a plain white stomach. We record their snout to vent length, or SVL, which is the kind of standard way of determining the length of the lizards, which is because they can drop their tails. So this is the kind of the best way to compare body sizes of lizards across all kind of health metrics, as well as the mass of the lizard would be collected just in grams with a drop scale, which can be kind of combined with snout to vent length to get a general body condition estimate to determine how much mass they have for how long they are. And then we would conduct our behavioral trials in the arena. So that end where the lights are tilted down is where the UV is higher, and the end where the lights are tilted up is where the UV will be lower and eventually reaching to zero. So we had a total of 56 lizards, and the first part of these trials, well, so before they could go into the arena, in order for the arena to function properly, as I was mentioning earlier, they have to have the UV sensor attached to their back. So that's what detects the UV while they're in that arena. But before they could even go in the arena, we had the first treatment of when the parietal eye was exposed. So we used a Q-tip to just apply water over the parietal eye and let it dry for about 15 minutes. And that was just to simulate the same act we would be doing to cover up the parietal eye later in the research. And that took about 15 minutes to dry, and it would take about 15 minutes for the next step as well. But essentially, once we did that, after the 15-minute acclimation to the placebo treatment, they would go into the arena for about an hour, and the computer that the sensors were hooked up to would record their selected UV value every 30 seconds. And then at the end of the 60-minute trial, they would be brought out and they would be given a chance to rest until the following day. And so the following day, we would apply non-toxic white paint to the parietal eye over their head. And that took, again, 15 minutes to dry. And then they would go back in the UV gradient arena, where for the same duration of trial and the same frequency of recordings. And so this is kind of how we would be able to compare these two treatments to see what the parietal actually does. And then once this had kind of concluded, they would just be released the next day after they had completed their laboratory trials. And so essentially the data we got from doing these trials was the average selected UV from each trial for each lizard, and as well as the set point range of UV from the trials. And so as far as the results we get from this study, they are pretty interesting as we'll see. So in regards to the kind of primary treatment of whether or not just covering the parietal eye has any influence on the lizard's not thermoregulation, UV regulation. We've seen our results from this study demonstrate that it does not, or we find no significant difference between the regulation. And so the covered treatment and uncovered treatment were not significantly different and are relatively stable. So in general, when you cover the parietal eye, there's no clear difference in our results that there's a difference in accuracy of regulation. And what that suggests to us is it's not enough to rule out that the parietal eye is not involved in UV regulation, but it does tell us that those other mechanisms I had mentioned earlier, like the lateral eyes or potentially something on the skin, that those are likely sufficient in terms of detecting the in terms of detecting the ultraviolet light they experience in the context that they need to regulate it. And so moving forward, the UV set point range, those middle 50% of values that they had selected, also had no impact whether the parietal eye was covered or uncovered. So just further supporting the results we have, the parietal eye had no clear impact on their accuracy of UV regulation. Now, where things get interesting is when we combine those results with the body condition of the animals. So we're mentioning the kind of length of the lizard being conjoined with the mass of the lizard to kind of score their body condition overall, how much mass they have relative to their kind of size. And so with this in mind, we see that the kind of larger lizards, those ones with the higher, more favorable body condition, had a high preference for higher UV values. And essentially what we kind of predict this corresponds to is to facilitate more thorough digestion if they've had larger meals or even more recent meals or are stockpiling nutrient reserves within their bodies. And potentially that would apply to both male and female lizards, but it is also possible that gravid females as eggs would also increase their mass relative to their length, which would look like a more favorable body condition, that it could also be related to gravidity or the egg development in females. But essentially, the trend we see here is just that the larger individuals prefer higher UV values. And this kind of holds true for the SVL values as well. So essentially, we see that when lizards were covered, the larger SVL lizards preferred higher UV, whereas when they were uncovered, they were at slightly lower UV. And essentially, when covered, if there is any disorientation potentially caused by covering the parietal eye, it would essentially just be possible that they are kind of hedging their bets in going for the higher UV value to be safe, maybe get a little more than their kind of true preference. And a smaller lizard might be selecting slightly less UV than typical, as there might be a greater risk of achieving too much UV when you are overall smaller bodied, and a greater risk of having too little UV when you are a larger bodied lizard. And then the other kind of avenue where Body size came into play was in comparing it with the sex of lizards. So between males and females, there was a significant difference in how they had regulated their UV. So the male lizards had the larger individuals selecting significantly higher UV values. And part of the advantage we see to this was, as we were talking about those displaying behaviors that they do to kind of court the females, would enable more activity and would facilitate more activity and potentially make them stand out more. So in order to stand out for those displays, you have to go in the areas where there's more light, and that kind of shows off their displaying attributes of their mating styles. And the females have a little more consistent of a mating preference because they're not under the same pressures to go out into the well-lit areas and show off for the other sex. And so with these results in mind, we kind of look forward to the future directions of where I think this research could go, or what could be done next with the kind of parietal eye and with UV regulation as well. So it's important to consider other modes of UV detection. And this is an area that I think is particularly relevant to understanding the adaptive significance of lizards in this context, and potentially the conservation of lizards in this subject, and just a better understanding of their evolution. So in order to understand how they regulate UV, it's important to know what they're using to detect it, whether it be the parietal eye or their lateral eyes or something else. And so if as UV and temperature levels both change in the kind of coming climate change that we're going through at the moment and for the foreseeable future, whatever whatever sensory systems they're using to sense the UV that's kind of incoming is potentially under subject for evolutionary pressures, and potentially just the things like that, and differences in their kind of evolutionary trajectory based off of how robust those systems are in the lizard species currently. It is also important, as more lizards are brought into captivity for things like assurance colonies, and understanding if a lizard's not regulating UV properly, is it a flaw in the UV we're providing, or is it a potential health problem in terms of what the lizard is able to detect, is able to receive to inform its regulation? And so in addition to the kind of parietal eye, the lateral eyes have also been demonstrated to have photoreceptors in the back of them that can detect UV, and there is the potential that they might have some ability to detect it in their skin as well, since that's where the cholesterol, where that gets converted to vitamin D3, actually is before being distributed elsewhere in the body. And so we've largely focused at this, what happens if you cover the parietal eye, but I don't exactly know how you do this, but in the future, it would be interesting to kind of see what happens if you cover all these potential methods of all these potential modes of UV detection. So like I said, we focus on what happens if you cover the parietal eye, but what happens if you cover the lateral eyes as well, since these also can function in UV detection. So unfortunately, we don't have lizard sunglasses quite yet, but if someone were to invent that, that might create a interesting operative study in terms of where they can detect the UV. And so another interesting kind of avenue of where this research is going, I think, is the body size of the animals. So we used a much smaller sized lizard than the previous parietal eye research, which yielded pretty different results to what they were seeing in regards to thermal regulation. So body size might have an influence on the role of the parietal eye. It might have an influence on the roles of UV regulation, as we were seeing larger lizards within this study, within that species, were choosing larger UV. And that is potentially a trend that we see at the whole, where larger species in general may want more UV to accommodate for the functions that go on in their body at that larger scale. And because adult lizards are larger than smaller lizards, it was also worth kind of looking more into the different age demographics and how these preferences of UV or the sensory capacity of the parietal eye differ throughout the lizard's entire life scale. And so, like I was saying, those previously studied lizards are much larger than the ornate tree lizard. We had sacrificed that kind of ease of blocking the parietal eye that they would have had in favor of a larger sample size. And so even the collared lizard in that group, as I was mentioning earlier, it's the smallest of them all, but it is still pretty large. They're actually large enough to eat the tree lizard that we used for our study. And so when we talk about the context of maybe a larger size or higher body condition being the driving factor of getting UV, these tree lizards would be that larger meal that drives that in a larger species. And so even regardless of size, just the amount a species eats, their kind of metabolic rate could be a determining factor in terms of UV and how their sensory systems operate accordingly with that in mind. And so the effects of reproductive strategies on the sensory capacity for UV or just UV regulation in general is another kind of interesting route to go down. So we had mentioned the tree lizards will do those kind of elaborate visual displays of doing push-ups and showing coloration, whereas something like a monitor lizard would do a little more of a slowly kind of tracking down with chemosensation and battling other males rather than just full-on displaying to appeal to females. And so there's different requirements for light if you want to show off color than if you just need to track down females and battle males. And so these kinds of different strategies in terms of reproduction and life history are another potential route where these sensory systems and these environmental pressures could differ. And then one of the more interesting kind of final notes on the future direction is the diel cycles. So the parietal eye is associated with those structures in the brain that produce melatonin. There's been some evidence that the parietal eye itself can influence the cycles throughout the day that each lizard goes through. And temperature and UV do have their peaks within the day. And as the climate changes, the time of day where their preferred temperature and their preferred UV are may become more stratified. And so being able to see what kind of things they prioritize at different times of the day using what sensory systems is, I think, a very interesting avenue for this research to continue moving forward. And so in general, what we can conclude from our study that we had done is that the effect of the parietal eye on UV regulation still at large remains fairly uncertain. So it didn't influence our results of selecting higher UV values, which is still interesting, because again, it might still be involved or it might not, and they just have other sensory systems that can compensate in the event that it's hindered, such as when the lizard's shedding or maybe covered in dirt or debris. And it is also possible that the parietal eye isn't used for these kind of short trial UV cycles, and that maybe it is just more geared towards longer diel cycles in order to inform the lizard's regulation ability. And so then the other conclusion we have is that the larger and more favorable body condition lizards prefer higher UV, and that within just males, that trend continues as well, where the larger individuals prefer higher UV exposure, which could be a further avenue to look down at the kind of more intricacies of diet selection and the diversity of species used in these studies, to kind of see how not only UV changes with these body sizes and the preference of UV and other environmental factors with these body sizes, but the way sensory systems are kind of used and prioritized. And in general, we can come to the conclusion that this is kind of, as long as research has been going on, you know, a lot of those studies were from the 1900s, late 1900s. We're kind of just getting started with really understanding what the parietal eye does, and we're certainly just scratching the surface at what UV regulation looks like in a diversity of lizards and the modes by which they can detect it. And so just to kind of give some more formal, longer references for the studies of parietal eyes that we mentioned throughout the talk, they're all listed here. And I would like to acknowledge the support that this project got from the Latanzio Lab at Christopher Newport University with my PI, Dr. Matt Latanzio being a big help for the project as well, and the various funding that CNU was able to provide me through the honors stipend and the ORCA student research grant, as well as funding just provided by the Organismal and Environmental Biology Department, and of course, the fellowship provided by the Audubon Appleton-Whittell Research Ranch, as that was certainly a big contributing factor to making this work possible, and of course, presenting it to everyone today. And overall, the biggest takeaway I want everyone to have today is to keep loving this vast diversity of lizards that we have, and to keep pondering why they all look and act and think and sense the world the way that they do. And with that, I will thank you all for your time and for your attention with this talk.
Thank you so much, Marcus. That was great. And thank you too. I think anyone that knows me well knows that my biggest secret at Audubon is I'm more of a reptile guy than a bird guy. So I really appreciate you giving me the excuse to think and talk about lizards a lot this field season. So I'm gonna throw my screen back up there real quick. There we go. And if it's cool with you, Marcus, we'll jump into a couple of questions.
Yeah, I noticed some in the already typed out in the question and answer box, so I can address those. What did we feed the lizards? We just use mealworms acquired from the kind of local pet stores in the area. And so they're appropriately sized for the lizards. They're easy to obtain in large quantities, and they're easy to just drop in the enclosures and let them eat them at their own leisure. And how did we remove the non-toxic paint before release? That's a good question. So it's just a water-based tempera paint. So essentially just dabbing it with a wet Q-tip after the trial removes all the paint. Sometimes the upon coming out, so they do like kind of thrash their bodies upon us picking them up. And so sometimes if the paint was dry enough, they would be able to flick off the paint scab on their head. But in general, just wiping it off with water was sufficient for removing the paint. Yeah, good question.
We'll give folks a couple minutes to drop any more in there that they might have. But I have one for you as well. One of the most interesting things I've learned from y'all at the Latanzio lab, not just that lizards use UV light to help navigate their environment, but that you've found situations in which lizards will put themselves in conditions of heat stress for the sake of UV regulation. Do you see any connections between that new tidbit of knowledge and the warmer, drier conditions that we are likely to see in the future due to climate change?
It's a good question and like a very interesting like topic and rabbit hole to just kind of dive down. I think a lot of it depends as well on how much the UV is expected to change in addition to the temperature as the kind of climate moves forward. So the structure of the atmosphere, how that differs, how much of the UV still gets through, whether or if more potentially gets through. And I think it would be different across different habitats, just in general, as a lot of the structural aspects that kind of when we're thinking about the atmosphere at large that we don't often think about, will also kind of influence that. So like canopy cover and the amount of tree cover is a good factor of how much UV gets through because it doesn't go through the canopy cover or the trees very well. And so in addition to climate change, we also have to worry about how much we alter the structure of the habitat, whether it's deforestation or just kind of advancing urbanization. And so it's a very interesting question, but unfortunately, I don't know how simple it is to answer at the moment, at least for me. So
Yeah,
it would
it's an
definitely,
interesting,
yeah.
just thinking through how critters react to and adapt to
Right.
our shenanigans is
Mhm.
a complicated one.
Yeah,
Cool. We have one more in the chat. Did you test the UV transparency of the white paint?
it's a good question. So in general, it's not another kind of thing we had noticed within our time at the Latanzia lab is it's not particularly difficult to block UV. Most opaque surfaces can do it. So in regards to the like surgery done for the previous methods, I can imagine that's relevant for if you want to control temperature regulation. Because just because you block the light, the parietal eye might have thermosensors around it, and if it's still detecting that temperature, it might not be as different in the lizard's behavior as you would think. Aluminum also has the ability to kind of reflect the heat from the light as well, with as you notice with how you will wrap your food in aluminum foil. But in general, the paint has been proven to be opaque enough to not let any UV get through it for the short periods of time we were getting. So like UV can degrade things like paint and plastic over time, but that's not really happening in the hour-long trial. And the paint was also white, so that's going to reflect some light as well. And that's the difference between using a like white paint, which kind of matches the reflective qualities of like aluminum, versus using a darker paint that would potentially, if it weren't opaque enough, would just be drawing more light towards the eye. So that was kind of the thought process regarding that, but that's a good
Makes
question.
sense. Well, those are all of the questions I see. Feel free to drop any more in while we close this out and we will address them. But I do want to just quickly mention, excuse me, Marcus's talk today was the final of 3 fellow-led webinars that we conducted this season. And all of them are available on the Research Ranch website. So just researchranch@audubon.org. Head over to the fellowship page and you will be able to find our 2 previous recordings, and we will put Marcus's up in the next couple of days once we have it ready. Oh, hey, there's another one. Do
Oh, yeah.
geckos, mostly nocturnal, have a parietal eye?
That's another really good question. And so it's in general, it's in lizards at large and within the kind of amphibians as well. So it's not always obvious. So that just because we can't see it doesn't mean it's not necessarily there, or that it's not there at some point in their life. But to level with you, I don't recall ever seeing a picture of a gecko with a parietal eye on its head.
Yeah,
Um,
me neither.
so potentially not. Potentially, that could be a thing they may have just lost with that nocturnal lifestyle. Geckos are weird enough as it is already. So I certainly wouldn't be surprised if they had lost it in that regard. But I don't recall ever seeing a picture of a gecko with its parietal eye exposed. But again, that doesn't mean they don't have it at some point within their life. But that is a good question as well. I'm now thinking about just the potential of fossorial species or nocturnal species having those kinds of modifications of whether they have it or not. But that's some good
Fodder
food
for
for thought.
future questions.
Yeah.
Right on. Well, I think that is it from the audience. Thank you again, everyone, for joining us. And of course, thank you, Marcus, for being here. We look forward to having you in the Latanzio lab out on the Research Ranch again, hopefully in the near future.
Mhm. Thank you.
Right on. Well, have a great rest of your day, everyone. Thank you again.